So technologies are fundamentally important and enabling. And as Sidney Brenner said, you can have ideas, you can make discoveries, but unless you actually have the technology available to answer the question, you won't be able to answer the question and make the discoveries. So, for example, PCR was a total revolution, changed everything how we did human genetics, how we analyze and sequence the human genome. The current revolution, and we're living through one at the minute, is obviously AI and all sorts of technological advances, targeted proteomics, untargeted proteomics, mass spectrometry. We live in an age that I always say to the students is 100 times more revolutionary than PCR in 1986, because everything, computational, molecular biology, and our capabilities in the lab have really exploded in the last 10 years. As I mentioned, we're very interested now in neuroinflammation, and we've been applying the central nervous system, the CNS NULISA panel, and it offers, again, that sensitivity, which is crucial, but it also offers, for example, modified forms of phosphorylated tau. And now phosphorylated tau 217, FDA approved, it's now an approved biomarker of Alzheimer. And that's a breakthrough, because trials now, or early experimental medicine studies, can be directed at lowering the amount of that protein very early in the disease process. And again, you want the most sensitive assay and the most specific assay that NULISA offers. So in that respect, that's the future, is condensing those key proven predictive biomarkers into small plexes of NULISA, and they can be quantitative, which Olink and NULISA are working on. I'm very happy at relative concentrations at the minute, because we're always comparing patients and controls and progression of disease. So I can see these technologies, their accuracy, their sensitivity, their quantitation, ending up at the bedside as routine predictive tools to molecular define disease, so that when drug companies like GSK want to figure out how to intervene in a pathway, they're actually intervening in the causative mechanism before the disease is entrenched and it's too late to treat or reverse the disease and its symptoms and conditions. So at the minute, NULISA is my favorite platform, and they're developing, and so are Olink, larger panels. I think the singleplex or 3 or 4-plex is a very important development. We need to keep bringing costs down, and we need to keep expanding the panels, so that when the literature tells us of new important biomarkers that survive thorough genetic and Mendelian randomization analyses, that immediately an assay can be designed and deployed across many labs.